Monolayer cell culture models have been very useful tools for studying various diseases including cancer. However, these models fail to recapitulate the three-dimensional environment observed in vivo. This video will demonstrate the use of PuraMatrix, a commercially available self-assembling peptide gel, to encapsulate and propagate the ovarian cancer cell line OVCAR5. The cell suspension is mixed with the PuraMatrix, and cell culture medium is added to initiate gelation of the PuraMatrix. When encapsulated and cultured in PuraMatrix, OVCAR5 cells assemble into 3D acinar structures that resemble the morphology of micrometastatic nodules.
Hi, I'm Adnan Abi-Yusuf. I'm a research fellow in the lab of Dr. Tayyaba Hassan at the Wellman Center for Photomedicine at Massachusetts General Hospital. Today we're going to show you a protocol for how to encapsulate and culture an ovarian cancer cell line in a synthetic matrix called PuraMatrix. We use this model in order to evaluate treatment response for ovarian cancer in our lab. So let's get started.
The BD 1% PuraMatrix Rad16 peptide hydrogel has a total peptide concentration of 10 mg per milliliter. Prior to use, reduce the viscosity of the PuraMatrix by sonicating it in a water bath sonicator for 30 minutes. Following sonication, while maintaining sterile conditions, aliquot the 1% PuraMatrix solution into sterile tubes to simplify future experiments. Avoid the formation of air bubbles. If they form, simply spin down the sterile tubes containing PuraMatrix at 1000 RPM for 5 minutes. In this demonstration, we will plate 2 sets of 4 wells at 2.5 mg per milliliter and 1.25 mg per milliliter total peptide concentration, respectively. Prepare 2 master mixes of PuraMatrix peptide based on the number of wells you intend to plate, which is 8 in this case. Each master mix should be prepared at twice the desired final concentration of total peptide, as it will be diluted with an equal volume of cells during plating. To make the master mixes, dilute the PuraMatrix with sterile 10% sucrose and 13.3% sucrose to total peptide concentrations of 5 mg per milliliter and 2.5 mg per milliliter, respectively, to achieve a 2x concentration of PuraMatrix in 10% sucrose. Then aliquot 25 microliters of the master mixes into 8 individual tubes. The PuraMatrix master mixes are now ready for encapsulating cells. The ovarian cancer cell line, OVCAR5, will be encapsulated and propagated in this experiment. Prior to trypsinization, incubate cells with 10 milliliters of sterile PBS without calcium and magnesium for 15 to 20 minutes. Aspirate PBS and trypsinize a cell monolayer cell culture. Neutralize the trypsin by adding cell culture media containing 10% heat-inactivated fetal bovine serum. Use at least 2 milliliters of media per milliliter of trypsin used in the previous step. Next, transfer the cells into centrifuge tubes and centrifuge the cells at 1000 RPM for 5 minutes to create a cell pellet. After centrifugation, resuspend the cell pellet in 10% sterile filtered sucrose. Count the cells and spin down again at 1000 RPM for 5 minutes to remove trace amounts of salt found in the media. Resuspend the OVCAR5 cells in 10% sucrose at 2 × 10 to the 5th cells per milliliter so that the final cell count per well will be 5,000 cells in each well. The cells are now ready for encapsulation in the PuraMatrix. The following steps for encapsulating cells in the PuraMatrix must be performed rapidly. Add 25 microliters of the previously prepared cell suspension to the first individual tube containing 25 microliters of the master mix of PuraMatrix. Quickly mix by pipetting about 5 times up and down in the tube without introducing air bubbles. Then pipette the 50 microliter cell suspension PuraMatrix mixture into the appropriate well of a 96-well cell culture dish. Initiate gelation of the BD PuraMatrix by gently pipetting 150 microliters of cell culture media to the side of the well. Repeat the steps for encapsulation of the cells for the remaining wells. Remember that these steps must be performed quickly, and the entire encapsulation procedure should be completed for one well before moving on to the next. After all 8 wells have been plated, leave the cells for 30 minutes. After 30 minutes, using a 200 microliter pipette, place the pipette tip in the well so that it does not touch the PuraMatrix bed, and very gently remove media from the well. About 2/3 of the media must be removed to equilibrate the pH of the hydrogel. Do not use a vacuum aspirator as this will disrupt the matrix. Next, very gently add 150 microliters of fresh cell culture media to the side of the well containing cells encapsulated in PuraMatrix. In the same way as shown previously, remove 2/3 of the media and add fresh media to the remaining wells. Replace the cell culture media in each well every 48 hours with fresh culture media. OVCAR5 cells were encapsulated as demonstrated in the preceding protocol. Representative 3D renderings of images of OVCAR5 cells encapsulated and cultured in PuraMatrix on days 1, 3, 5, and 7 are shown.
So we've just shown you how to encapsulate and culture the ovarian cancer cell line OVCAR5 in a synthetic matrix called PuraMatrix. So when using PuraMatrix, it's important to realize that this protocol has been optimized for the OVCAR5 cell line. Each researcher will have to optimize and fine-tune the protocol to their particular cell line. And remember, when doing media changes, it's very important to not disrupt the matrix bed as it's very fragile. Never aspirate and always use a pipette. So that's it. Thanks a lot for watching and good luck with your experiments.